Lecture 2, Part B. So when I ended the Part A of this lecture, I had described to you how protein kinase A mediates epinephrine's flight or fright response. This line of reasoning is true for most receptor pathways in the G protein class, and so there are over 1,000 G protein coupled receptors. They activate about 4 different classes of G protein subunits. Heterotrimeric G proteins, which consist of 3 subunits, an alpha, beta, alpha, and a beta gamma. They're essentially called heterotrimeric, but beta and gamma are always tightly bound to each other, so they're functionally heterodimers, if you want to call them that. So the alpha subunit defines both receptor specificity and often effector specificity. Or very often effector specificity. The effectors are enzymes, channels, or sometimes even regulators of small G proteins. Occasionally, but not always, they produce intracellular second messengers such as cyclic AMP, the most classically characterized one, IP3, inositol phosphate, another soluble second messenger, and a membrane-bound second messenger, DAG or diacylglycerol. Activates a variety of protein kinases, such as protein kinase A, protein kinase C, protein kinase B, calcium calmodulin dependent kinases, cAMP kinase 2, and these kinases in turn mediate various physiological effectors such as transcription factors, metabolic enzymes, and so on. So this is pretty much a linear signaling pathway, if you want to call it that. As you can see, the signal flows down a set of coupled biochemical reactions, but even in this linear signaling pathway, in its simplest form, there is almost always a feedback loop where the protein kinases phosphorylate receptor to either most often inhibit them, a situation called desensitization, where upon occupancy by the ligand of the receptor gets turned off for further stimulus for a period of time. So in addition to these large G proteins or heterotrimeric G proteins, there is a very big family of small GTPases, which are about 21 to 28 kilodaltons in size. The typical large G protein is about 90 kilodaltons in size. The alpha subunit is somewhere 40 to 45 kilodaltons, and the beta gamma another about 42 or so kilodaltons. The small GTPases are another class of signal transducers, which also allow for signals to flow from receptor to effectors by which allows signals to flow when the signal transducers are activated when GTP is bound and inactivated when GDP is bound. In this little schematic, I show you a typical cycle, GTPase cycle for the small G protein Rho, which is involved in cytoskeletal regulation. Many receptors couple to these small G protein pathways, not just the GPCRs that couple to the heterotrimeric G proteins, although they are a class 2. Here the small GPCRs are called serpentine receptors. So in addition to the GPCRs, cell adhesion receptors such as integrin, cytokine receptors, growth factor receptors all couple to the small G proteins, and here the example used is the RAS pathway, a very famous oncogene, and they all couple by regulating exchange factors called GEF, guanine nucleotide exchange factors, that allow the small G protein to be activated from the GDP bound state to the GTP bound state. And as I told you, the GTP bound state allows for signal to flow from the receptor to effectors. And then there are these proteins called GAPs or GTPase activating proteins that are involved in the active in the active deactivation of the GTPases, converting them back to the GDP bound state, and this sort of gives rise to a cycle. Since many GEFs and GAPs. There are many GEFs, there are many GAPs, there are many protein kinases, and all this mix and match stuff go on to produce an extensive network. So the regulation of GEF and GAPs are a sort of an important point at which networking occurs, and we will discuss this more in the next lecture. Here, let me move on from signal transduction or the basic process of signal transduction to sort of focus on receptors. You can see that because signal transduction pathways involve signal flow, they are major targets for drugs for a variety of diseases. Receptor ligands are widely used as drugs. That's because you just need to get the ligand into the bloodstream and they can bind to the receptor at the outside of the cell surface. Agonist receptors, ligands that bind to the drug and initiate biological action are called agonists. Receptor ligands that bind specifically to the receptor but do not initiate action but block the deleterious effects of natural ligand are called antagonists. And in between the agonist and antagonist, there is a class that can be called a partial agonist, or if you want to call it, it's also a partial antagonist. So these partial agonists or antagonists will specifically bind to the receptors, but they will not fully activate the receptor. So consequently, by themselves, they produce, say, 30% or 50% of the activity, and then they're partial. But now if used with a full agonist, in excess, they bind to the receptor and suppress the activity of the full agonist, and so they might behave like partial antagonists. So these receptors can come in many classes, and common drugs that are receptor ligands, the best known of these are insulin that binds to the insulin receptor, which is an agonist drug used to treat type 1 diabetes. There are, of course, very famous antagonist drugs, such as propranolol, which is an antagonist for the beta-adrenergic receptor, was among the first hypertensitives to be developed to lower blood pressure, and for the development of propranolol, Jim Black shared a Nobel Prize back in the 80s. He also invented or designed and developed cimetidine, which is an H2 receptor antagonist that blocks acid secretions. These are small molecules and is used for a treatment of GI disorders. There is, of course, drugs against tyrosine kinase receptors, such as the antibody trastuzumab that binds against the tyrosine kinase receptor ERB2, and this drug is used to treat certain types of breast cancers. So, in both biochemistry and in pharmacology, mathematical representation of drug action has been a major way in which drug action is studied and understood, and it's used for the development of drugs because you want to develop more potent and efficacious drugs. So basically, coming back to this ligand receptor interaction reactions, one can indeed have a ligand receptor interaction reaction that can contain both activators and inhibitors, which is the most common kind of scenario where the natural hormone or the neurotransmitter might be the activator and the drug that is given exogenously might be the inhibitor, and then you can have reactions going in both directions, and the ligand receptor for in the formation of the ligand receptor complex, which is the sum total of the complex that can transmit the information, is given by this equation down here. This type of equation can be used to determine the extent of the physiology or the pathophysiological reaction, and also both the extent and the action of drug action. So the typical kind of experiments that were done in the 70s and 80s and all was to have a radioactively labeled ligand, which could either be an agonist or an antagonist, and then you would have excess amounts of the unlabeled ligand, and one could generate a series of binding curves of the ligand to the receptors, which one could observe by counting the amount of radioactivity that's bound to the receptor, and in the presence of varying concentrations of the antagonist would produce a set of curves that look like this, set of curves that have varying sort of time courses. From calculating the apparent affinity constant for the receptor of the labeled ligand in the presence of excess unlabeled comparator, one can get a series of, say, affinity values or observed KD values, and these can be plotted as a function of the concentration of the competing ligand, and this plot on the right hand side, when extrapolated down to the ligand concentration axis, gives you the relative affinity or the affinity of the receptor for the antagonist. This is an important value, and this value can be calculated in various ways, because in designing a drug one wants to get the highest affinity possible, because this allows one to take smaller quantities of the drug, and typically higher the affinity, the more specific the interaction can be. The take-home points for the lecture 2 are as follows. Signaling pathways receive information from the outside of the cell and change cell physiology in response to this information. Indeed, they change sometimes tissue and organ physiology and organismal physiology, and all the processing and change in information can be represented mathematically. And this mathematical representation is very important not only for understanding biological function, but also in drug development. Signaling pathways contain many components, each of which receive and transmit signal with bidirectional specificity. Just to emphasize this point, let me use the examples of GS and GQ. GS couples to GS coupled class of GPCRs and activates adenyl cyclase in response to these receptor activation. GQ11 proteins, in contrast, couple to GQ11 class of receptors and activate phospholipase C in response to receptor activation. GS does not activate cyclase, GQ does not activate GS does not activate phospholipase C, GQ does not activate cyclase. So there is specificity at the effector action as there is specificity at receptor action. So this bidirectional specificity of many components in line allows for signaling pathways to emerge from reactions of a component with an upstream and downstream partner. Information flow through signaling pathways can be studied mathematically using ordinary differential equations. So this has been done a long time. This is not something new for systems biology. This has been done for a long time in both biochemistry and pharmacology, and adapting these sort of biochemical and pharmacological lines of reasoning into systems biology helps us build bigger and bigger systems that can be computationally analyzed to understand input-output relationships and how information is processed as it flows to the systems. And of course, receptors are major targets for drugs that are used to treat a variety of diseases, and understanding drug action requires understanding both receptor affinity or drug affinity for the receptor and efficacy of the drug, and these are rooted in sort of mathematical representations and computations of receptor function. This concludes lecture 2. Thank you.